Thanks for bearing with me. And thanks so much to AIMOS as well for this really cool opportunity. It's so great that the society is able to do this and yeah, support early career researchers. So in the short time that I have, I thought it would be a good idea to just share one snapshot of one of the projects in my PhD. Okay, so as you all probably know, in psychology, we've been going through this replication crisis. And that's led us to reflect on our research practices and to ask ourselves, how can we better ensure that our findings are accurate? So some of the topics that have been discussed, at least in psychology, have been the prevalence of false positives, sometimes called type 1 errors. And that's how often do we think we've found an effect when actually there's no effect there? And there's compelling evidence that suggests that these are probably more prevalent than we think they are. Statistical power, when we're studying true effects, statistical power is how often we'll be able to detect them. And power depends on things like the size of the effect we're studying, our sample size, and our pre-specified type 1 error rate. And lastly, p-hacking, which involves making data-dependent decisions to make results look good or statistically significant. Okay, so because of all of this discussion that's been happening, we were interested in studying how these might affect results that have been published in social and personality psychology articles. So are the results that are reported in this literature consistent with a literature that's full of low-powered studies, a lot of p-hacking, and maybe even both? Or are they consistent with a literature that's full of pretty decently high-powered studies? And more importantly, in light of all of the discussion that's been happening, have these things been changing over time? So when we're studying true effects, we'll get smaller p-values much more often than we'll get larger p-values. So this is a plot of the distribution of p-values for a simulated data set. And all of the studies in this plot have 80% power to detect the effect that's actually there. There's also no p-hacking or false positives included in the simulated data. On the x-axis, you can see the range of p-values all the way up until the conventional threshold in psychology, 0.05. And on the y-axis, you have the frequency of those p-values. And as you can see here, at the start of the x-axis, where the p-values are lower, with 80% power, these smaller p-values are much more frequent and much more common than the larger p-values at the other end of the x-axis, which are much less common. So now I've added a red line, which reflects the p-values in a simulated data set where each study has 50% power to detect all of the effects. Again, all true effects, no p-hacking, no false positives. And you can see that power decreases and the slope gets less steep. So we see fewer small p-values and we see a few more of those larger significant p-values. So as statistical power decreases, this line gets progressively flatter, and it's quite easy to simulate different levels of power. But to go back to my question, what happens when we add p-hacking, false positives, variation in power, and the interaction between all of those things into the mix? Well, the shape of the p-curve starts to be affected in very complicated ways. And I'm not going to explain what happens there in this talk because I only have 15 minutes and because I honestly don't quite know how those things would change the shape of the p-curve. It's all very complicated. For the sake of simplicity, what I'm going to do today is model the real data against those two simulated lines that I just showed you to compare whether the shape of the p-curve is in the real data is changing over time and to see whether it might be consistent with either of those two scenarios. So these were the two social and personality psychology journals in our sample, as well as the timeframes that we studied. They're both pretty mainstream traditional journals. And that first one, JPSP, is the flagship journal of the field. It's pretty high impact. And we also only looked at one particular section of that journal, the attitudes and social cognition section, which is mostly experimental studies. In the second journal, SPPS, that's a relatively high impact journal, but slightly less so. And there's also a wider range of social and personality psychology topics in that journal. So a few key events that are worth pointing out on this timeline. In 2011, you have the infamous Feeling the Future paper by Darrell Bem, claiming that ESP, extrasensory perception, is real. And that was one that Annie mentioned earlier. And that was actually published at this journal, as I said, in 2011. And the famous false positive psychology paper that coined the term p-hacking also came out around this time. So there was quite a bit of turmoil following this, and we often refer to that as the replication crisis, with concerns about replicability rates, among other things, growing. And of course, a lot of things have been happening since then, but since I only have a short amount of time, I just wanted to point out one more relevant thing in this time frame. And that's that an open science advocate, Samir Vazir, who's just sitting over there, became the editor-in-chief at SPPS in 2015 until the end of the time frame that we studied in 2019. And Samir brought in some progressive changes, focusing on making statistical power and statistical reporting a priority at the journal. So that certainly changed the core values of the journal there. But it's also a conflict of interest on our part, worth stating, because Samir was involved in this project and involved in checking the articles that we coded that were published at SPPS. Okay, so to code articles, we started by creating a list of all of the articles that were published at each journal in each year. Then we went in and we scanned each article to check if there were any experiments, and we included quasi-experiments as well if they used experimental language. And then we read each article and we extracted a key hypothesis from the article. And we located the key tests that mapped onto that key hypothesis and the test statistic block, among other things. And we did that for hundreds of articles. And we used those test statistic blocks to recalculate a p-value for each experiment. And then we plot the distribution of all of those recalculated p-values into a plot that looks something like what I showed you before. And in our case, we're also going to be plotting that real data against those simulated curves that I showed you, the 80% and the 50% curves. And this is really as a guide for what sort of slope we might expect to see under certain levels of power. But these would really be the best case scenario for the data we're seeing. And once you account for false positives and for p-hacking, the curve would probably start to get a bit flatter. So this is how many articles and experiments we could code. And for the results that I'm going to show you today, the sample size is at the level of the articles that we could code because we randomly selected one experiment for each of these articles. So finally, to visualize change over time, we had to bin the years that we studied so that we would have at least 2 periods within each journal that overlap so that we could look at change over time between the 2 journals. And we decided to plot separate p-curves for each of the time periods that are up here. So we have a very early period in JPSP, 2 overlapping periods that are highlighted there in blue, and then 1 later period in SPPS. So let's jump into the results for JPSP. So the black line here represents data from the early period in JPSP right before the catalyst of the replication crisis really started. And it seems to mostly be falling below that red line, which represents underlying statistical power of 50%. So the best case scenario here is that this curve is looking worse than the world where all of the studies have 50% power and there's no p-hacking, no false positives, meaning that we can detect effects half of the time. You can also see a bit of a bump in the black line of p-values that are around 0.04 to 0.05, which could be indicative of p-hacking. So overall, this doesn't look great. I've also included the median sample size there to give us a look at how this is changing over time as well. And you can see that there were about 70 participants per experiment. That was the median. Let's see if things improve. So looking at 2010 to 2012, this was right at the start of the crisis, with the Feeling the Future paper and the false positive psychology paper coming out in 2011. It doesn't look like there's a lot of change here, but given this is just prior to and at the beginning of the crisis, it probably makes sense that we're not seeing much change just yet. All right, and in the final period between 2013 to 2015, there again doesn't seem to be too much change, but the median sample size per study does seem to be increasing, which is a positive sign and is also consistent with a lot of meta research that we're seeing that suggests that sample size and power are increasing. So have things changed at JPSP? Well, we can probably rule out the possibility that there have been big changes on this front over these years at JPSP in this section, but we need more evidence to really discern how small that change is and whether it might still be meaningful. We also don't know whether things have changed in later years and after the replication crisis has had more of a chance to have an impact on this literature. And so that is where the second sample comes in. So in SPPS, things look very bleak at this journal right at the start of the replication crisis. That black line looks almost close to flat. So we're seeing an almost uniform distribution of p-values with not many more smaller p-values and larger p-values. And that's concerning because to get this kind of shape, it could be that there's a lot of false positives in this sample. There are a lot of really low powered studies. There's a lot of p-hacking or probably some combination of all of the above. We also see that the median sample size here is 84 per study. In the next time period, things look less bleak. There's some more of those lower p-values and slightly fewer larger significant p-values, but that black line is still below the red line, which reflects, again, 50% power. So there's a lot of room for improvement. And then between 2016 and 2019, something kind of amazing happens. And so now the black line is more closely following the blue line, which represents 80% power. And the median sample size has almost doubled from the last time period. And that's a really stark and surprising jump. So maybe things have been changing in this area on social and personality psychology. Rationalizing this post hoc, you might remember that I mentioned before that Samin, who supervised this project, became the editor-in-chief at this journal in this time period, and statistical power and statistical reporting were really a priority there. So an explanation that seems really likely to me is that this jump that we're seeing is the result of a sweep of more progressive editors at SPPS. And that might mean that rather than reflecting a broader, more robust change in the field, this could really just be change happening at this one particular journal at one point in time. But it would be really interesting to see how these results compare to that of similar journals in the late 2010s to see if this is really a trend that's occurring at the field level. We also don't know whether this trend will continue in future years with the latest editorial team, but it's definitely an interesting question. So have things changed in social and personality psychology? Before I answer that, there are a few important limitations of this study that are worth keeping in mind. So first, it's important to remember that we only looked at this one particular metric. And of course, there are lots of other ways that these journals could have changed. Many of these articles, particularly for SPPS, were coded by just one person, which makes it less likely that errors could be caught. And we also had to make decisions that were sometimes subjective about the key hypothesis, and sometimes those decisions had to be made ad hoc. And last, interpreting the shape of the p-curve is not straightforward. So our simulations were overly simplistic and overly optimistic. There is a great paper by Stefan and Schönbrut with simulations that suggest that p-hacking strategies can actually lead to a right-skewed p-curve even when none of the studies included are studying true effects. And that means that a steep right-skewed p-curve could be the result of A, bunch of really well-powered studies, B, some really skillful p-hacking, or C, some unknown combination of all of the above. And those are some very different explanations for the data that we're seeing. So we spent 8 years collecting this data, and what have we learned? Mostly, I've learned that p-curving is incredibly hard. So the process was time-consuming. It's often hard to tell what author's key hypothesis was, and sometimes they don't even report a result that maps onto that hypothesis. Also, the inferences that we can draw from the results of p-curve turned out to be not as straightforward and more complicated than we originally thought. In terms of findings, we found some interesting descriptive patterns, and we were quite surprised at how much the shape of the curve changed at SPPS in that last period, which we take to be a good sign for those articles. But it's complicated, and there are a lot of limitations that mean that we can't draw strong conclusions here. But with all that being said, I'd be curious to hear your thoughts. Thanks so much to Adrian and to AIMOS for, again, the scholarship and the chance to speak here, and for making all the people that make this possible.
Hi, everyone. I'm happy to be here today. I received the top of scholarship award to support my thesis, and I successfully defended my thesis in June of this year. Today, I will share with you my thesis project, the rationale, objectives, our approach, procedure, and some selected results. Psychology is a discipline that strives to understand the human behaviors. Unfortunately, psychology mostly investigates people from the US and Europe, ignoring a population like Africa, home to like 17% of world population. For example, if we are to understand moral issues in psychology and ignore Africans, it will create an unknowns situation, meaning we will not know what moral issues exist in Africa. These thoughts and other issues are the underlying reasons for my thesis. First, the questionable research practices, the notion that researchers engage in contentious research practices in an attempt to find support for a false hypothesis. Scholars have argued that this poor replicability rates of psychological science claim can be linked to the questionable research practices. For example, hypothesizing after results are known. Also, in psychology over rely on the US and European samples and theories. Most research interest samples and authors are from North America and European population. This image about the dominance of US from Collett and Saston paper shows that about 80% of the sample used in papers published in some evolutionary psychology journals are from North America and Europe. In fact, Africans are underrepresented in global research. African population, like I said, represents 70% of the world population. Still, the African sampled use in studies published in top-ranked psychological science journals ranges from 0 to 4.5% of that. In my thesis, we argue that this underrepresentation is linked to generalizability problem in psychology because we find it increasingly difficult to apply most psychological science claim beyond the US and Europe. And also the generalizability problem is linked to the replication crisis that we are familiar with. So the thesis objective broadly is to improve generalizability of psychology, specifically to improve African research capacity in open science practices and victim science, also to test the applicability and heterogeneity of psychological claims within and outside Africa. And lastly, is for us to recommend intervention on adopting open science and victim science and improving African participation in psychology. Our approach, like in the first phase, we identify and train African colleagues in open science and victim science, replication-like studies. Some of the tools are the open science framework. In the phase 2 of this thesis, we conducted a collaborative replication and education project. These platforms allow you to train people in replication studies while they adhere to open science practices. For us, we use this to build African collaborators' capacity in open science and also prepare them for a larger study, and that is the many-labs Africa study that examines the extent to which African effects are replicable and generalizable to population from Africa, from Europe and North America. And also this study helps us to build African researchers' capacity. My presentation is focused on the many-labs Africa study, so I will go into that fully. We executed this many-labs Africa study in 2 parts. One that is a preparatory work, where we consult and we conducted a consultation, and we also conducted a study selection. Then there is the main study, that is the replication study. For the consultation, we consulted with researchers working with the Global South population to determine the type of study to replicate, and we determined that we will replicate applied social issue studies that are previously investigated as a practical application to African society. Second is for us to determine an approach for selecting the study. Contrary to the most popular large-scale cross-cultural replication studies that we've seen that select convenient or popular Western claims, we adopted like a 3-stage bottom-up that's heavily driven by local researchers. Our study selection is in 3 stages. The first stage, African researchers nominated research area and interest and actual study to replicate. In the second stage, we collect this information, we get this information and we search for studies. We evaluate the nominated studies and we shortlisted 5 studies. In the third stage, we now go back to all the collaborators from the target study population to assess the shortlisted study for final selection. To ensure that we select African studies that are reproducible, we adopted criteria categorized into three. One, the locality criterion. For example, we are interested in studies that are of interest and relevant to African collaborators. So studies that are feasible for multi-site online study. We rely heavily on studies that use less complex design. The third criterion is that we look at for studies that reported p-value that are less than 0.01 and that are well documented. I can share results from the study selection, a very interesting results. If in the figure are assessment results to select the final 3 studies from the 5 shortlisted studies. We conducted this assessment among our African, European, and North American researchers. On the x-axis are the average values, and on the y-axis are the 5 shortlisted studies. In each panel are the dots represent the continent average value, with Africa in red, Europe in green, and North America in blue. Overall, what we observe is that the preferred studies polarize into Africans versus Europeans and North America for interest, relevance, and applicability to their population. From the figure, you see that the extramarital affair and the teenage pregnancy, you can look at the first panel that I have with ranking. So the marital affair and the teenage pregnancies stand out as the African preferred studies, in contrast to European and North American assessors preferred self-disclosure, civic engagement, and condom use negotiation studies. So for the main study, ends because we see the dichotomy between African and their European and North American counterparts. We prioritize and selected Africa preferred studies. So our final study are one, the perception of teenage pregnancy, originally investigated among Nigerian parents. 2, gender differences in attitude towards condom use, originally investigated among Ghanaian adolescent college students, and willingness to forgive extramarital sexual relationship, originally conducted among Mozambican women. I will skip studies 1 and 2 for the want of time and present the hypothesis and results for study 3 only. Our focal effect for the study 3 were the main effects of emotional involvement on willingness to forgive extramarital affair. 2, the regrets expressed on willingness to forgive extramarital affair. We use 8 scenarios that describe a husband involved in low versus high emotional involvement in extramarital sexual relationship, and 2, if they express regrets, low versus high regrets. Participants responded to what extent they are willing to forgive a cheating husband on a point scale of 0 to 10. Our target sample are the original sample used, and these are women. To collect data for all these studies now, we administer the studies as a bundled study plus demographic questions, and it's sexual orientation measure for exploratory purposes. We randomize the presentation of the measures and items in each measure. The site sample their local general adult populations via social media, online research platform, and on sites using anonymous study links. We're able to collect data from 43 sites, 18 countries, and over 5,000 participants. We used a mixed effects model to analyze, to examine our effects, and this effect on 18 focal effects among the subgroups sampled in the original study, and also whether Africans differ from North Americans and Europeans. So my results will just be the one for the study 3, the extramarital sexual relationship study. Now for the results for study 3, because the p-values were smaller than the preset alpha, we determined that we replicated the effect of emotional involvement and regrets expressed on willingness to forgive a cheating husband. Just in this figure, we have 3 panels for the 3 regions: Europe, North America, and Africa. On the y-axis are the average ratings for the willingness to forgive, and on the x-axis are the level of regrets expressed by a cheating husband. In each panel are the red and blue crossbars and dots for the low and high emotional involvements, respectively. The crossbar are the original average values, while the dots are the replication values. So what we observed that the original mean scores were larger than the replication mean scores, and also the claim generalized more to Africans than to Europeans and North Americans. In fact, the 2 claims replicated for the extramarital affair study are 2 of the 4 out of the 18 African originated claim replicated. Overall, African originated claims generalized more to Africans than to Europeans and North Americans. Region comparison showed that the focal effect sizes differ between Africans on one hand and Europeans on the other hand. The heterogeneity checks show that these findings do not vary between the sites across continents, even for sites within the same continent. Lastly, our failure to replicate 14 claims could be as a result of the cultural differences and some of the issues that we suspected before, the questionable research practices. On a final note, I just want to say that we have some way forward. I suggested in a recent book on a better how to research in the Global South. For example, I suggested that we need more infrastructure for improved producibility for Africans, and we need to adopt our open science practices for credible research. Thank you for your attention.
Hi, my name's Emma, and I'm a final year PhD student at the University of Edinburgh. I work with the Camarades Research Group. I'm supervised by Emily Senna and Peter Kind, and my research is funded by the Simons Initiative for the Developing Brain. My work involves systematic review and meta-research of biomedical animal studies, and I'm going to be saying a little bit about what I do and what I found during my PhD. My PhD is focused on neurodevelopmental conditions with a single gene cause that are typically characterized by intellectual disability, seizures, and autistic traits. These include Fragile X syndrome, Rett syndrome, and SYNGAP1 related intellectual disability, to name a few. These are studied using animal models that share alterations to the same gene that's implicated in the human condition. Animal research is a key part of the drug discovery pipeline. This includes basic research, which is used to understand the biology underpinning aspects of development, degeneration, and disease, and preclinical research, which is used to test novel therapeutics to establish safety, efficacy, mechanisms of action, and side effects. When we're making decisions about or relating to animal research, we need to ensure that we consider and evaluate all available evidence. Systematic reviews are one way of summarising and evaluating this evidence. In animal research, they can be used to improve experimental design and animal welfare, identify gaps in the evidence, examine potential sources of heterogeneity, and inform research decision-making, such as what drugs to take forward to clinical trials. In project 1 of my PhD, I wanted to conduct a review of systematic reviews, essentially to identify systematic reviews that were already published, looking at animal models of genetic neurodevelopmental conditions, and assess their reporting against a draft extension of the PRISMA guidelines called PRISMA Pre. This was a registered report, which I got peer-reviewed through peer community and registered reports. From the review, I identified 12 relevant publications. 10 of these were peer-reviewed articles and 2 were conference abstracts. And what I found was the reporting quality across all of these publications was mixed. Many methodological details were missing, and few reviews conducted risk of bias assessments or linked to a pre-registered protocol. From this, I could see that despite a growing number of resources designed to help researchers conduct systematic reviews of animal data, these just kind of aren't being used when people are doing systematic reviews. And if you'd like to read more about this work, this has recently been published in Brain and Neuroscience Advances. Systematic reviews are incredibly time and resource consuming, and simply keeping pace with the published research is difficult. So in project 2, I sought to answer the question, how do we identify and use research when there's so much published research, it can be hard to find anything? To answer this, I implemented a pipeline of software tools to generate a high-level overview of research using animals to model genetic neurodevelopmental conditions. This involves running a broad, systematic search at regular intervals to capture new research as it's published, training a machine learning algorithm to identify the publications that are most likely to be relevant from the search results, and tagging these relevant publications with features such as reporting of measures to reduce risk of bias, open research practices, and experimental details. In the end, we can summarise all this evidence and display it in an interactive and searchable web application. If you'd like to read a little bit more about this approach, you can read our paper, which was published in Clinical Sciences last year. The web application is still under development, but this is a screenshot of one of the app pages, which shows a number of relevant publications identified. As of last month, almost 15,000 publications were identified. We could see that 12 of these were marked as retracted using information from Retraction Watch, and other aspects of the app include visualizations of different data from these publications and a searchable database where you can download the results as well. Some of the key findings from this project were that human validation of the data is key to ensure that data within the app is reliable. Machine learning will miss some relevant publications and it will include some irrelevant ones. Additionally, poor or vague reporting in the papers makes it hard to extract different features. Missing metadata relating to publications was also a key issue. A number of publications did not have a DOI, which meant I couldn't retrieve full text or OpenAlex data, and a lot of publications were missing data on things like funders, which limited the usefulness of certain data summaries and visualizations within the app. When performing text mining, I found the PDF to be a really inaccessible format, and I kind of hypothesized that more structured text formats like XML or HTML are more likely to be helpful for this kind of text mining work. My third and final project aims to show a use case of the database I developed in project 2 and assess its utility by conducting a systematic review and meta-analysis using the app and traditional methods that's relevant to researchers in the field. I worked with animal researchers to come up with a research question for the review, which asks which factors account for heterogeneity in SYNGAP1 rodent seizure phenotypes. I'm currently working on this project. The review protocol is under review at Prospero. Traditional searches have been complete, but not the app searches yet, and the screening is underway. One of the key overall findings from my PhD has been the need for synthesis-ready research and data in order to feasibly conduct systematic reviews and other forms of meta-research on animal data. Information from a study, so the study's data, and about a study, the article metadata, need to be reported and available in ways that enable their reuse. A key guidance for researchers would be to report experiments clearly, such as referring to the Arrive 2.0 guidelines and making experiment protocols available and sharing well-documented data. My guidance for publishers and database providers would be to consider the utility and provide more open metadata and make machine-readable text formats available for text mining and analysis. And that's a summary of my PhD so far. Thank you for listening. Thank you to my supervisory team and collaborators, and especially thank you to AIMOS for awarding me the top-up scholarship and allowing me to present my work here.
Hey, good afternoon, or in my case, rather, good evening. My name is Max Primbs, and as you can probably gather by the fact that I'm presenting here, I was also one of the AIMOS Top-Up Grant Award winners. I do love my random lotteries. Now, today I'll be presenting a many-analyst investigation of a many-lab study on gender-inclusive language. To begin, I just want you to close your eyes for a moment. And I want you to imagine a politician. Whose picture shows up in your mind? Who's the first person that pops up in your brain? Is it maybe this guy? Or is it maybe this lady? Maybe after the last election, not so much anymore. Now, if I were to play a slightly different alteration of this, if I were to ask you to imagine a male politician, maybe he comes to mind again. He's hard to get rid of, like a cancer. But maybe also Barack Obama comes to mind. Or for the Australians among you, Peter Garrett. Who would most likely not come to mind is Kamala Harris. And why would she? I explicitly ask you for a male politician. Likewise, if I ask you to imagine a female politician and name her, Kamala Harris might come to mind, or Hillary Clinton, or for the Europeans among you, even Angela Merkel. Who for once, hopefully, would not come to mind is Donald Trump. Now, in the English language, a lot of words that refer to groups of people, like politicians, are inherently gender neutral. And if I want to refer to a subset of this, I need to use an adjective like male or female to specify if I want politicians who are men or politicians who are women. But that's not the case in every language. A lot of languages, such as German, Spanish, Dutch, or Italian, are inherently gendered. What that means is that there are different word forms for female and male versions of the same word. For example, for the word politician, the male word form would be politica. So if I ask you to imagine a male politician, I would ask you to imagine a politica. Likewise, if I would ask you to imagine a female politician, I would ask you to imagine a politikerin. And I promise this unwanted German lesson is almost over. The last thing I will teach you about German is that if you refer to a group of people, for example, multiple politicians, imagine Donald Trump, Kamala Harris, Hillary Clinton, and Angela Merkel standing in a little circle, you would use in German the male word form. So these four people together would be referred to as Politiker, even though 3 of those 4 people are women. This is called the generic masculine, and it's inherent in many gendered languages. If you refer to a group of people, and there's even 1 man in that group, or there could potentially be a man in that group, you use the male word form to describe this group of people. Now, several research studies over the past few years have investigated this generic masculine, and they have found that it biases the cognitive retrieval of exemplars. What that means is that if I ask you to name 3 politica, you will most likely name more men than women. Probably even 3 men. Now, this cognitive bias induced by the way German language, but also Spanish or Italian language works, biases downstream representations and has consequences for hiring and promotions. If you think, for example, which engineer to hire, and you use the male word form, it might be that the people who mostly come to mind are men. So the chance that a man is hired is much more likely. Now, scientifically, this has been investigated for the first time in 2001 by Stahlberg and colleagues. And they found that for different groups for different social categories, so for politicians, for athletes, for singers, and for other groups, the use of the generic masculine means that more male exemplars come to mind. Because of this bias, people developed several gender-inclusive word forms. And Stahlberg and colleagues also compared the generic masculine to these gender-inclusive word forms. And they found that the gender-inclusive word forms promote the cognitive retrieval of women exemplars. For example, if you want to refer to a group of male and female politicians, you could just use the male and the female word together. So Politiker und Politikerin. This is called the feminine masculine. What you could also do, and what people started doing in writing, because Politiker und Politikerinnen is fairly long, is to use the so-called internal I form, which uses the male word stem and then adds the ending of the female word with a capitalized I. Now, there are also people who do not feel captured by the gender binary, and for those people in the past few years, a gender star form was developed. Now, a lot of schools and universities and even some state governments started to increasingly use these gender-inclusive word forms. And that very quickly became a topic of societal debate in Germany. The Association for German Language argued that these inclusive word forms are inherently agrammatical and violate German rules of grammar. They also argued that they make texts more difficult to understand, and particularly for people without a background in higher education, or for people who do not read a lot of texts, it makes it hard to follow a text. Therefore, they argued that these gender-inclusive word forms should not be used. On the other hand, scientists and a lot of young activists wanted to make German language more inclusive, and therefore promoted the use of these word forms. Now, this culture war recently came to a culmination with one of the biggest German states banning the use of gender-neutral or gender-inclusive language in schools and public bodies. That means if you work at a school or a university in Bavaria, which is also the state of Germany I am from, yay, we're like the Texas of Germany, you are not allowed to use gender-inclusive language. And Bavaria isn't the only state which had this fantastic idea, and other states are trying to implement their own laws, trying to ban gender-inclusive language. Now, because of this escalating culture war, a group of researchers thought that we really need better and high quality evidence for the negative effects of the generic masculine. Evidence that is much harder to deny than a study from 2001, which comes with all of the issues and problems studies in 2001 had. For that reason, a group of researchers led by Hilma Bromer decided to conduct a many-lab replication and extension of Stahlberg and colleagues' original paper from 2001. Now, Bromer and colleagues in this many-lab replication decided to test 3 different hypotheses. In the first hypothesis, they simply replicated the original hypothesis by Stahlberg and colleagues. And they hypothesized that the internal I and the feminine masculine form, which I showed you earlier, would yield a higher number of female exemplars mentioned compared to the generic masculine. Now, in hypothesis 2, they sort of expanded the pool of forms used, and they added the star form, and they also added a neutral control form. And again, they expected that the gender-inclusive forms would yield a higher number of women mentioned. They also had hypotheses about the base rate effects, but these are less important here. And they managed to collect 2,700 participants across 12 cities in Germany. And while this paper was going through the review process, one of the journal editors, Hans Eisermann, put out a call asking if people were interested in doing a many-analyst investigation of this many-labs replication. And that's how this many project I'm presenting here today was born. Now, together with Shilin Neo and Marina Orefici, we collected 150 analysis teams who were all willing to run their own version of these analyses and to investigate the 3 hypotheses on screen here. Now, what did we find? Well, this is a forest plot, and originally we planned to have 3 forest plots in total, 1 forest plot for each hypothesis. Turns out doing a many-analyst investigation is much more complicated than we thought it would be, because different research teams interpreted the hypothesis very differently. Some research teams reported total effects of all gender-inclusive word forms compared to the generic masculine. Others reported individual contrasts. Here, for example, on the screen, you can see the total effects, comparing both the feminine masculine and the internal I form to the generic masculine. And what you can see is that most odds ratios are greater than 1 and do not include 1 in their confidence interval. That means that across all analysis specifications, there's evidence that use of gender-inclusive language facilitates the retrieval of women exemplars. Now, other research teams looked at individual contrasts. And the orange bars here are the feminine masculine, and the green bars are the internal I. So you can see that the internal I was much more efficient at promoting the retrieval of female cognitive exemplars. Now, for hypothesis 2, the star form and the neutral control form were added. And people went absolutely crazy on the contrasts. So some teams compared the total effect of all gender-inclusive forms to both the generic masculine and the control form. Others investigated the individual contrasts of all 3 gender-inclusive forms compared to both the generic masculine and the neutral control form. Other teams, again, did the contrast but only compared it to the generic masculine, while a 4th group of teams only compared it to the control form, and there are just too many forest plots because people did so many different things and investigated the hypothesis so differently. So I will just skip that for now and skip to my main takeaway, which is that gender-inclusive language increases the cognitive retrieval of women exemplars. And it does so across many, many different interpretations of the hypothesis, across many different model families and analysis specifications. I also sort of have a personal takeaway from this, but this is less positive and more negative. If you look at this and if you look at family, you will see that it says linear for most. And then if you look at the confidence intervals, you will see that they are very large and a lot of them span towards the end of the graph. And they span towards the end of the graph because they reach infinity. And a lot of teams led by PhD level researchers, so postdocs or assistant professors, submitted analysis where confidence intervals reach to infinity. And this just doesn't sit right with me. This should trigger your bullshit sensor as a scientist, but apparently it doesn't. And that just makes me very concerned for the state of social psychology and for the skills of some of the researchers in it. Anyway, to end on a more positive note, this was a collaboration with Marina Arefisi from the University of Cologne and Xiling Neo, and it was funded by the Utrecht University Open Science Grant and the AIMOS top-up grant. We are very thankful for the support. And we also had an amazing research assistant called Joop Speelman, who deserves a lot of the credit because he actually was the one who did the data analysis in the end. Because I'm not here in person, because I'm old and it's late, I want to thank you. And if you have any questions, you can reach me on Twitter, Blue Sky, or per email. Thank you very much.